At the moment then, we've got the blood has passed into the glomerulus under high pressure, and the liquid and, well, the water and other small components of the blood can be squeezed out. Similar way to forming tissue fluid, but this is a little bit more extreme. The pressure difference is more between the vessels coming in and out of the glomerulus. You've got the podocytes supporting the capillaries so that they aren't damaged by the pressure. And you've got this filtrate that appears in Bowman's capsule then, that's got all sorts of useful stuff in it. And it then starts to make its way along the tubules. So if you follow the route on the diagrams that you've got, the next step is obviously the proximal convoluted tubule. So we're going to talk through that a bit. There is also some reabsorption of ions by the distal convoluted tubule, but it works pretty much the same way. So I'm not going to deal with that as a separate item. And obviously there are going to be thresholds really that will determine how substances are reabsorbed. So, you know, do you take back all of the substance that's been removed, or do you take it back to a certain point? And it depends. It does vary. So some ions will be completely taken back, others will be only taken back to a certain concentration. Glucose will almost all be taken back as long as we're under normal circumstances, but if there's a huge amount of glucose in the plasma, perhaps because somebody can't control their blood glucose level, and therefore, you know, they're not putting it into storage, so their plasma glucose is very high. And if you're doing the assignment choice on glucose control, then that's something to consider, because plasma levels will rise really high. It will pretty much all be squeezed out into Bowman's capsule, but then as the filtrate then passes through the rest of the tubule, it's not all taken back up, because there's a threshold level above which the blood can't take the glucose back up. Hence, you get glucose in the urine of people with diabetes, uncontrolled diabetes, I should say. So glucose in the urine is a sign that the level in the plasma is so high that the kidney can't actually take back everything that was squeezed out. So for the different substances, we're going to have different thresholds, different outcomes. We also, of course, have to reabsorb water, but that's the separate bit. That's coming up on osmoregulation. Now, the bit that I've got on selective reabsorption in the PCT is complex, but I decided I would just put it in and explain part of it. And the yeah, you can see by the number of points on this slide that it's yeah, slightly more elaborate than perhaps we would ideally want. I think I'm going to just look at this slightly back to front. If we look at the processes that are going on first, so down at the bottom of this slide, we've got some things that we've met before. We've met active uptake, that's like active transport, so it's just a matter of pumping certain substances back. We've met passive uptake, that's diffusion, simple or facilitated, depending on the size and the charge of the particles, remember. And of course, endocytosis we've met, you know, certain substances might be reabsorbed by that method. The one that doesn't look familiar is co-transport, but in fact we have met that. Co-transport that we've met, it can actually be facilitated diffusion, but we've met an example that was active transport, and I don't know if you can remember, but where two substances are involved in the same transport mechanism. Nervous one, yeah, which was moving. The sodium-potassium pump, that's right. So you'd got sodium going out, you'd got potassium going in, but it was the same transport protein that was doing it. So that's an example of co-transport. Sometimes, and in this situation, you've got transport of two substances, and they sort of influence each other, but glucose and sodium ions can be taken back up by a co-transport process. I've realised I've managed to get a superscript bracket round my plus at the end there. In order to carry out these processes then, what you have in the proximal convoluted tubule is a different type of adaptation structurally to what you'd got in the Bowman's capsule. Bowman's capsule, we've got things like the podocytes and all that. In the PCT, we've got cells that are adapted in different ways. So if we have a look at the list here, tight junctions between cells to ensure transcellular movement. I think they're actually called tight junctions as well. That's not just, you know, an everyday description of them. They are known as tight junctions, where there's unlike the capillaries in the glomerulus, where we're saying that there are little pores in the wall, so substances can move really easily between the cells, therefore it's possible to filter out no end of stuff. Here, in the PCT, you can't just let anything pass between the cells because they've got these tight junctions. And so what has to happen is the substances actually have to move through the cells. So that's what transcellular movement means. So in the PCT, the cells are adapted to ensuring movement of substances, reabsorption actually through the cells themselves, the tubule walls, all right, rather than squeezing in between any cells. The cells in the on the inner surface of the tubules have microvilli, which are little tiny extensions, and they also contain a lot of mitochondria for ATP, for the active transport we've got down here on the processors. And the basal membrane is the outer layer of the tube where it's going to come into contact with those peritubular capillaries. And you have the membrane that's sort of attached to that is folded a lot, so you've got quite a large surface area there to allow substances to pass into the blood. Now, you have got a diagram of this. I've got a really rubbish one on a slide. I'd forgotten that you've got one in your handouts. The top right one on page 389, the textbook bit that we were looking at before. All right, diagram in the top right-hand corner. All right, the top left-hand corner, you've got your ultrafiltration diagram. You've got the glomerulus, the Bowman's capsule, podocytes and all that, and they're really nice little diagrams there. But the diagram next to that, figure 8.17, reabsorption in the proximal convoluted tubule, that's actually no end better than the diagram I've got on the next slide. But you can see that the cells there, you can see they fit all these descriptions, and you can actually see something labelled as a tight junction on there, and it's a little sort of, you know, bit that sticks out that actually makes a tight junction with the next cell. You've got the microvilli on the surfaces down the right hand side of the diagram there, the little fingers sticking out to increase the surface area. You've got mitochondria in there, so that matches up. And then the basal membrane, the left hand end of those cells, you can see it's folded in, so you've got a large surface area for substances to move out. And then you've got your blood capillary running down the left hand side, and you can see substances being moved through there. You've got glucose and amino acids coming in through the microvilli, but then finishing their journey through the intercellular space that is to the left of the tight junction at the top there. You've got some sodium ions being taken in just sort of directly through, sort of diffusing through, I think. Water coming in there as well. Water is not a significant reabsorption in the PCT. You've got some urea being absorbed. Well, how useful is that? Not at all, is the answer. But because you end up with a concentration difference, there will be diffusion of urea back into the blood at this point. And this is a bit of a problem that we have to then sort out later. So consequently, we'll finish this whole section off with secretion, where the blood actually secretes. Blood vessels will secrete stuff they really want to get rid of, and that'll include urea, because it just won't stay out of the blood. It's a bit like James's assignment and the box, you know. It's you got rid of it, and then it's sort of it's back in there again. And then yes, you've got a few proteins being taken up by pinocytosis. That's the endocytosis process that we've got on the slide there. And so that's actually quite a useful diagram to summarise that, and I would say that is a heck of a lot better. I couldn't find one like that, and I'd forgotten to refer to the handout, so I ended up putting this up there. You can see that's actually at some point been copied off a book, and it's wonky. It's just slightly off horizontal, isn't it? You should see it better than I to see on the screen. It's not straight if you just follow the words across the top. And I find that really disturbing. I don't like it, so I'm probably going to lose that slide now I'm reminded that there's a decent diagram in your handouts. But again, it was showing, that you've got the, I suppose I can use it to identify microvilli here. All right, so these are your microvilli. This is the tight junction. It's not labelled as such on there. And then the bottom edge of these cells has got these infoldings to increase the surface area. You've got your mitochondria for energy, for active transport. So it's all about adaptations of the cells for taking up glucose, amino acids, sodium ions. We haven't got everything else on here that's actually in the diagram in your handouts, so that is definitely the better diagram. There is water. You will get some taken up just purely by osmosis because of the concentration difference. Having removed a whole lot from the blood, you're going to get it taken back, aren't you? So that's what happens in the proximal convoluted tubule, and it's very important to us because we can't afford to lose all the stuff that's been squeezed out in Bowman's capsule. But the water uptake that we get there is not enough. There is some water reabsorbed in the PCT, but I think the estimate is if we lost all the water that was squeezed out in Bowman's capsule, we would dehydrate to the point of death within probably less than 5 minutes. All right, so we have to take back pretty much 99%, I think it is, of the water that we've squeezed out. So it does go back in. However, we do also need to lose a bit of water because a lot of the waste products that we're producing and needing to lose are soluble, so they need to be washed out. Although that's almost a backwards way of thinking. Animals that need to retain more water will tend to produce more solid sort of waste products. And you get, for example, birds, for example, they don't produce sort of separate solid and liquid waste, do they? They produce a sort of in-between sort of combination. Sorry? Lizards the same, yeah. Reptiles and birds obviously sort of quite closely related. So it's about what they're excreting as well. And if you've got plenty of water passing through your body, as you might have in a fish, say, then you've got no problems anyway, and you can actually get rid of substances like ammonia that's really soluble in water, which is a bit hard for us because we do lose some ammonia, but it's so toxic we can't have it hanging around, and urine production is urine release is a little bit slower in us. So this whole system has to be modified. It's one of the sort of, I suppose, most obvious systems that would be modified in different environments. So, desert animals, aquatic animals, that sort of thing. So, remember the kidney. Right, let's now move on to the loop of Henle. Now, if you've got, this up here just says osmoregulation in the medulla. Right? If you've got some of your diagrams for this, it will probably help. I can't tell you which ones are the best on this, but if we've got any... Oh, yes, there is actually, nearer the end, there's the handouts that start off where it says biofact sheet. The kidney, excretion and osmoregulation. This was just my first port of call, really. The page 3 in that is actually focusing on osmoregulation. All right, and there's a diagram of the loop on its own there, the loop of Henle. The numbers on there may or may not match up with the ones I'm going to use. The actual numbers are not critical. Oh no, we haven't got numbers on that one. Okay, I don't know whether I've got another one that has got numbers on it. In terms of concentrations, I'm thinking, no, we haven't. That's all right, that's fine. Okay, how the heck are we going to get all the water back then? We squeeze out most of it, but we need a lot of the water. So we squeeze out most of the water in the plasma, to be honest. We need to get most of that back. And if you look at the medulla, of course, arranged in its pyramids, the things that look like seashells on your diagram, then what we've got on the left here is representing a section of that sort of shape. So you have the wider bit at the outside of the kidney coming to the narrower bit at the bottom, because all the Bowman's capsule and the convoluted tubules are in that wider part, and then most of the narrow part, you've got the loop of Henle and the collecting ducts, and they need to be brought fairly closely together because they are the bits that are really responsible for reabsorbing water back into the blood. So the cortex around the outside, cortex around the outside is going to be the capsule, the Bowman's capsule and the convoluted tubules, and then the medulla is mostly collecting ducts and loops of Henle. Not entirely, there are some that appear in different places. Okay, so we now need to have a look at the process. Sorry, this is a little bit complex, but it's not so bad actually. I've given myself plenty of time to work through this. We have this thing called a countercurrent multiplier mechanism. Okay, so up here, countercurrent multiplier mechanism. Countercurrent just means that you've got flow in opposite directions. So with the loop, you've got the two limbs. The filtrate goes first of all into the descending limb from the proximal convoluted tubule, loops around the bottom, and then comes up the ascending limb before it gets to the distal convoluted tubule. So you've got down on one side, up on the other side. So that's the countercurrent. That's all that means. I shouldn't make my hands go up and down like that. So they're going in opposite directions, and it's a neat sort of evolutionary result here. What it's able to do is to create a concentration gradient along the length of the loop. If you have flow going in the same direction, I think I just need to sidetrack to fish. No, hang on. I don't know that I've got a. No, I haven't got a smartboard open, have I? If you have two vessels running side by side, and you've got a high concentration in one and a low concentration in the other, water will move from the low concentration to the high concentration, but that could mean that halfway along your two vessels, they've got equal concentrations and there's no more movement. So they've reached an equilibrium, and you get no more movement, I'm thinking water here obviously, along the rest of them. If, however, you have them flowing in opposite directions, although you don't get such a big gradient between any 2 points on those two vessels, you actually have gradients all the way along, because in one vessel you have the highest concentration coming in at one end, but with the other vessel you've got the lowest concentration coming in at the other end. So as they go along, the one that came in with the highest concentration is gradually going to get less. The one with the lower concentration is gradually going to get more, but they don't equalise because you maintain a sort of small gradient all the way across, so it makes it much more efficient. You don't really you'll never need to explain that to anyone, not on this course anyway. But the way this works in the kidney is that we've got this loop with its descending limb and its ascending limb. The descending loop then, I should have put descending limb on there. I think, that needs editing. It should say descending limb. Same with this one. Descending limb of the loop is permeable to water, but it's not permeable to ions. All right, now the ions we're talking about particularly are sodium ions. They tend to be accompanied everywhere by chloride ions. They always like to hang around together for obvious reasons. So the descending limb, you can't move any sodium chloride in or out of it. It's permeable to water, but not to the ions. On the other hand, the ascending limb, all right, the one that goes up on the right side of the diagram there, is permeable to ions, particularly sodium, accompanied by chloride, but not to water. So this gives us a way of actually setting up a difference between them. Now, what we get is, first of all, overall, sorry, I rearranged these bullet points for a reason. Overall, sodium and chloride ions are going to move from the ascending limb of the loop into the tissue fluid. And that's what we've got happening over here. All right, so we've got sodium and chloride just leaving that part of the tubule into the fluid, the tissue fluid that surrounds everything. Now, at the bottom end of the loop, down here, there's enough of a concentration difference for them just to diffuse out. But the further up the loop you go, because some has diffused out there, you've now got less of a concentration difference as you go up this side. So as you head up the ascending loop, you've actually got a harder job for diffusion. So in the upper part, if you come up here, and that's the bit of on the diagrams, is the thicker part of the tubule. The upper part of the ascending loop actually pumps ions out. So you get active transport up at that top bit. All right, diffusion is okay down here because that's where the highest level of those ions will be, because they've only just come around from this side. So there's going to be most of them here. They start diffusing out, all right, as we come up here. The concentration drops, so we're actually then at a difficult point trying to get them to diffuse out. So active transport is used to actually deliver them out higher up. But all you really need out of that is the fact that the ascending loop is going all out to deliver sodium and chloride ions out of the tubule and into the space between the limbs, you know, into the tissue fluid, the blue area on that diagram there. So what we've ended up with then is a concentration of those ions in that space. They can't get back in, let's say, to the descending limb because that's not permeable to sodium and chloride, only to water. But we've increased the concentration there in that area. So what's happened to the water potential? If we've pumped sodium and chlorine into it, its water potential will drop. Yeah, so we've reduced the water potential. It's now concentrated. That increased concentration or reduction in water potential means that water is going to be drawn out of the descending limb. So water comes out into this space to try and dilute the sodium chloride, and in doing so it ends up going back into the blood. Right, because it gets into that space, it then enters the blood, because the blood of course was really concentrated once it's come away from the glomerulus because it had all its water squeezed out. So it's sort of a way of tricking the water back into the blood, isn't it? It's not tricking it, but the concentrating the sodium chloride in that space is going to pull the water out of the filtrate, and once it's actually in the tissue fluid there, it's also going to move into the capillaries because they've got even more concentration in there because they lost all their water earlier on, or not all their water. All right, so this idea of the counter current thing then is the bit on this diagram that's confusing is the blood, but we've got the down on that side and the up on that side. That's the counter current. And if you look at numbers for this, and this is where it gets a little bit tricky. All right, if you look at numbers, what's happening? Well, it's what happens first. Really, you know, you don't have a starting point for this, to be honest. Although I suppose the best starting point would be as the fluid comes into the loop from above. So where they've got number 1 on this diagram, and you've got a concentration. I don't know if this is just sort of arbitrary units. We haven't got an actual measure for this. But number 1, we've got passive transport. And this is because the filtrate that's coming in at the top here is quite dilute. It's got it's got all the water in that was squeezed out in Bowman's capsule. Right? They've had very little taken back at this point, so there's a lot of water in there. So the first part of the loop of Henle will be losing water through passive processes, because there is going to be just more water here than there is in the surroundings. So if the filtrate's losing water, it becomes more concentrated as it heads down here for a start. So as it comes down, it's just getting more concentrated, but of course the passive process is only part of the story. On the other side, we have, down at the bottom, we've got the passive transport. Probably because they are charged particles, that'll be, well, what will it be? If it's passive but they're charged, what will we need? Well, passive would suggest what sort of process basically would be diffusion, yeah? But because we've got charged particles, it'll be, that's it, facilitated diffusion. So that's, yeah, likely to be facilitated diffusion. Couldn't be sure. Yeah, right. Again, that wasn't as hard as it sounded. So facilitated diffusion just from a higher concentration into lower. But then once you get up to this thicker part up here, where you've got the thicker walls here of the ascending limb, that's where the active transport is pumping out the ions. And you can see, you know, obviously the higher you go up, the more have been pumped out. So the more dilute it gets going up there. Now that means that on the left side of this diagram, your descending limb, it gets more concentrated as it loses water. But on the right side, it gets less concentrated as it loses ions. So that means that the lowest concentration here is next to the lowest concentration here. All right. But you've also got the highest here will be next to the highest here as well. We haven't got a number there. Of course, the very highest is down here, where it's lost as much water as it can, but it hasn't lost the sodium and chloride ions, so you can get your highest concentration down there. So this multiplies the number of, well, the length of the concentration difference. Right? It's not big. You can see at any point, you know, you've got 300 here to, well, 100 there. You've got 900 here to, well, whatever that would be, might be 800. You know, it's not huge, but it's enough, and it's all the way along is the important thing. All right, complicated system, but really quite amazing, isn't it? You know, in terms of how it works. And, you know, you have to wonder how something like a kidney evolves. But we are talking millions of years, aren't we? So, you know. Yeah, well, it's not the only organ we've got two of. And built-in redundancy, you know, so that if one's damaged, the other can take over. And partially because we develop in a fairly symmetrical sort of way. I mean, you can manage with one. Similarly, you can manage with one lung. Other than that, yeah, we're pretty much stuck with one of everything else, aren't we? It is amazing how you actually get to this point, isn't it? Yeah, you start with a fertilised egg, and that's just one cell. Divides into 2, then 4, then 8, 16, and so on. And then, yeah, ultimately, you end up with things that are as remarkably complex as this. I mean, if you study any aspect of zoology and evolution, you can look at particular organs and see what their evolutionary sort of pattern probably is. I have no idea what the route would be for a kidney, but for the heart, it's actually fairly well documented that you basically start off with, there are invertebrate animals like worms, for example, often just have blood vessels that contract. You know, just a single one of their blood vessels has muscle tissue in it that will actually help to drive the blood along. And that's it. It's not a heart with chambers or anything like that. And then you might get perhaps 2 blood vessels working together, or an enlarged area in that one blood vessel that makes it more effective at pumping. And you can see how that develops. But with a kidney, I just, you know, think the nature of the interaction between the blood vessels and the tubules It's just absolutely fantastic, and there's another reason for putting it back in, because it does fascinate me. Quite like the kidney, even though it's complex. You're not expected to be able to explain this whole sort of, where numbers and everything on here, but just get this overarching idea that you've got the descending limb and the ascending limb with different permeabilities. The sodium and chloride leave the ascending limb by active transport at the top and then passive diffusion, likely facilitated at the bottom. So you increase the concentration of the tissue fluid in between. That pulls water out of the descending limb by osmosis. That water goes back into the blood. That's, it's not that hard when you look at it like that. Okay, so in that case then, have we got any scope for sort of adaptation here? Well, we have. Mentioning animals that live in different environments. So, for example, if you've got an animal that needs to retain more water, because let's say they live in a desert, very dry conditions, what do you think might happen? I'm not giving anything away. Physically, camels have a slightly different approach to water management, but because they are usually given water to drink. If you think of desert, because they're domesticated, if you think of things like, I don't know, desert, sort of the rats and things like prairie dog type animals, animals that live down burrows, don't get a lot of water. What sort of adaptation could you make to this to absorb more water? No? You'd have a longer loop. If you make a longer loop, you can absorb more water, can't you? Because you've got, an even longer set of gradients. So it's, animals that live in desert conditions and have to retain, practically all their water as far as they can, have much longer loops of Henle than we do. And I'm assuming aquatic animals probably have shorter ones because they don't have issues really with retaining water in the body. So this does give us an area where we can actually make a modification that allows absorption of different amounts of water. Right, so that's the basic system. Now, in terms of the control of this, and we should just be able to fit this in quite nicely. Going back to the idea of osmoregulation and antidiuretic hormone, which we had right at the beginning of this unit when we were talking about hormones. I used ADH as one of the examples. And then you will remember aquaporins, won't you, from the osmosis assignment. And I did say I'd mention them again and introduce them because this is one place that they're quite important. But what we've got here is a really nice overlap between osmoreceptors, which are part of the nervous system, ADH, which is one of the hormones we mentioned, aquaporins, which comes from the last unit anyway, and collecting ducts, it says up there. And the control process here is quite a nice way of sort of seeing how everything coordinates together, really. And when we started, and we did ADH, I just said, if the concentration of your blood falls, then osmoreceptors detect it, or if it rises, the osmoreceptors detect it, and this will then cause the production of or stoppage of antidiuretic hormone. Now, a diuretic is something that increases urine production, so an antidiuretic reduces urine production and enables the body to hang on to water. So an antidiuretic effect is what you want when you want to retain water, because the blood's more concentrated. So what happens? Well, I think I've got two diagrams for this. I've actually, we'll see which one works best. But this one actually rather nicely sort of summarises the whole thing. The hormone itself comes from the pituitary gland. Remember, that's a little bit of the endocrine system that connects with the nervous system. And it works like this. All right, you don't get ADH until there's a dehydrated condition. Dehydration will mean less water, so it'll mean a drop in the water potential of the blood. And the way that that is actually detected, the way that the osmoreceptors work, which I think is quite neat, it's this, you know, this overlap between so many things. If the blood becomes more concentrated, it will draw water out of the osmoreceptors. They tend to shrink, and it changes the membrane potential of the osmoreceptors. They then start firing off impulses. So that's what we've got at the top here. Okay, this hypertonic up here, this is very concentrated. So water's been drawn out of our cell body of our receptor here, and it starts firing off the impulses, and that's what these are. All right, so those are nerve impulses. So the osmoreceptor is a nerve cell. It fires off impulses when its cell body shrinks because it's lost water. However, not only is it a nerve cell, it also secretes a hormone. Beautiful little overlap, and these are known as neurohormones, I think. Doesn't sound quite right, but neurohormones. And so we end up with the release of ADH here. I don't know whether that's directly from that or whether it's elsewhere, but you get an increase in the production of ADH, the antidiuretic effect. So antidiuresis results in returning to our set point, which is, you know, considered to be the healthy, the normal concentration of blood, if you like. And that will then just result in the cell having its normal diameter and firing off far fewer impulses. All right, only 3 there for, you know, about 8 up there, I think. On the other hand, if you get an increase in the water level of the body, so you get the hypertonic situation down there, so more water, less solute, it makes the cell body swell. All right, so we've got a much bigger one here. And as the cell body swells, we get fewer impulses. So I've only got an odd one there. I just realised we've got ion channels here that open. They will be sodium channels, won't they? I'm just wondering, these what we've got on here, this is a little plus. I think those represent sodium ions in there. So what happens when the cell shrinks is that sodium channels will open and fire off the impulse. Okay, when the cell swells, it looks like those channels are now closed, so we'll only get occasional impulses. Consequently, we don't get ADH produced. There's a reduction in that, and the body can lose water through the kidney as much as it likes. So that's the control system in terms of the hormones, but I haven't mentioned the aquaporins yet. Where the heck do they come in then? Well, they're affected by the ADH, all right? So ADH is going to affect the number of aquaporins that there are. So we've got the control system through osmoreceptors and the production of a hormone. If we then look at what does the hormone do? Well, ADH, hang on, I need to move that again. So, can't get it to click. Can't. All right, I can't move the let's try it down there. That's because I had the pen in my hand, I think. ADH is it's not affecting the loop of Henle. All right, now, you know, your immediate thought would be, well, you know, if we're talking about water, changing the water absorption, then loop of Henle is where that all happens. But that's a fixed system. This countercurrent multiplier, you can't change that with a hormone. All right, it's just about the concentration of the fluid coming through. So the loop of Henle tends to work in a very constant sort of way. The only thing that will affect that is the amount of water that's in the filtrate and the concentration of the blood. So it doesn't matter, you can throw as much ADH at that as you like, and it's not going to affect that, the countercurrent thing or the differences in concentration. Where you do get an effect is with the collecting duct, and the collecting duct runs pretty much next to the loop of Henle anyway. But ADH, what have I put? Increases the permeability of the collecting duct to water. It upregulates, increases, that's another word for that, production of aquaporins. So ADH is actually having an effect on the production of these proteins in the cell. It's increasing the protein production. And those water channels, then if you've got more of them, they are placed in the membrane, all right, as a result of ADH. All right, remember when we first did cell membranes, what do we describe the model of membrane structure as? It's the fluid mosaic, yeah? And that the fluid mosaic model means that it's not a constant composition, it will change. So we can put proteins in, we can take proteins out, we can change it, they can move about. And this is what ADH does. ADH causes more aquaporins to be put in the cell membrane of the collecting duct. If there's more aquaporins, more water can pass through. So you can actually get more water going which way? I've forgotten now. Passing from the filtrate back into the blood. All right, so it's water coming out of the urine, isn't it? If we want the anti-diuretic effect, we don't want to lose the water. So the increased number of aquaporins. So this means less water remains in the filtrate. The urine becomes more concentrated. The individual urinates less. That's what we call antidiuresis. And the water that's come out of that filtrate can then be taken back into the blood. So your little diagram down here, there's two parts to it. If the individual then is dehydrated, you've got the bottom part here where you've got the, let's get this the right way around, the little purple blobs here are the aquaporins. All right, so if you're dehydrated, you've got more aquaporins. The water can pass through those. If you're okay hydrated, you've got fewer aquaporins. And so this is where you've got the high ADH. All right, so ADH causes the production of more aquaporins. They're placed in the wall of the collecting duct, although it says tubule cells down here. All right, that's the wall of the collecting duct. And so if you've got more aquaporins, more water can pass out of the filtrate. All right, this is what would eventually go on to become urine. So more water is going to be taken back from that, and it ends up back in the blood over here. All right, so there's more water absorbed when the ADH is there because there are more aquaporins in the collecting duct cells, and so water can get back from the filtrate back into the blood. So that just joins together a lot of the things that we've actually done, if you think about it, in terms of osmosis and aquaporins and cell structure, membrane structure, hormones, nervous system, kidney. I mean, it's almost a secondary thought there. But if I'm going to go back to that previous one just to go over that again briefly. All right, so if you start at your set point over on the right hand side over here, the set point is normal water potential of the blood. Okay. If the blood gets more concentrated, all right, in other words, your water potential falls, the osmoreceptor cell body will shrink. That'll open sodium channels, so they'll rush in, fire off impulses. That will cause the production of ADH, and then the antidiuretic effect will reduce the water loss. And we now know from the last slide, the slide after this, that is by increasing the permeability to water of the collecting duct. But if the blood gets less concentrated on the lower part of this, then the cell body of the osmoreceptor will swell, the channels close, you get fewer impulses sent off, you get a reduction in ADH, so there is less water taken up, there are fewer aquaporins in the walls of the collecting duct. All right, so I think it's just a really sort of neat system, really control system here. So we've actually got two things involved in osmoregulation, and that's really sort of the focus. And when we get onto revision, I won't come back to the PowerPoint because that is there on Moodle, but I will do some revision on this on Notebook. All right, so you've got the PowerPoint on Moodle, you've got the narrated version of it. This is going to go on to YouTube. You've got the handouts, and I will do a revision bit on it anyway to flag up the bits that you need. So one of the things that we'll do during revision is to cull your handouts. All right, you can either create just a revision set from your handouts, or just put tags on them or marks on the ones that are useful, because we've done a lot more than you're actually going to need for the exam. So yeah, if this is all going a bit whoosh over your head, don't panic. All right. But you should be getting a real sense of, isn't biology amazing from this, really? The actual, how it can control all that stuff at this microscopic level is really quite impressive. Anyway, and finally, just to finish this bit off, which makes it quite tidy. So I'm just going to spend a little while on this now, very little. And then next time, Monday, we can overview the processes. We can look at the composition of urine, just overall. Perhaps look at some of the options for dealing with kidney failure. Come back to some more questions, and then start thinking about revision. Looking forward to that. The exam is still a fair way off, but obviously revision, Monday the 20th, the first Monday back after half term. So get it out of the way. Yeah, you've got yours on the Monday afternoon. The health group have theirs Tuesday morning. So if anybody sort of falls off on Monday afternoon, they'll be in doing it Tuesday morning. Right, now this last little bit, and this is without covering absorption in the distal convoluted tubule, because it is just really more of the same, to be honest. Well, not quite the same, but similar. In terms of processes, we had ultrafiltration at the beginning. We had selective reabsorption in the PCT. We've had osmoregulation. So this is another process that just helps to modify the composition, really, of the blood, is what we're looking to do, because blood goes into the kidney in one state, and you want it to come out all clean and with exactly the right concentration of everything. So what's happening is the kidney is modifying the composition of the blood, and then the blood obviously has a link with tissue fluids. So the kidney is controlling the composition of all our internal fluids, isn't it, really? And just to finish the story here, really, urine composition then is going to be affected by this process of secretion. And secretion is, a lot of it is active transport, but we've got two processes going on here. And this is secretion from the capillaries. The capillaries are actually making an effort here to get rid of stuff that we really don't need. So it's a secretion from the peritubular capillaries to the renal tubular lumen, I've put there. I don't know why, I must have copied it from somewhere. The lumen is always a space inside a tube. We have them in blood vessels, we have them everywhere else. So it's the space in the tube. I just could have put tubule, to be honest. So it's the opposite process of reabsorption. Reabsorption is taking back from the filtrate things that the body needs, whereas this secretion is, seriously making an effort and often expending energy to actually pump stuff out that we want to get rid of. So we've got two main processes. Both of these we've met before, so these in themselves are not a problem. Passive diffusion, movement of molecules from the capillaries to the fluid in the nephron. Not a, it will just be down concentration gradients, but the active transport part of it is actually using energy. So I've put pumps, ATPase. Just going to start introducing that. Probably should have done earlier on. When ATP is used for energy, you have to break it down, if you remember. And the enzyme that breaks it down goes under the wonderful name of ATPase. Ase just means enzyme. So ATPase breaks down ATP, releases the energy, and that's going to allow the movement of substances against the concentration gradient, of course. Now, the substances that are most commonly moved by this mechanism, some of these are more obvious than others. We're not going to look at any of the biochemistry here of this lot, but these are just examples of what the what the blood vessels might be secreting. Potassium ions have to be kept fairly constant because if you've got too many or too few, they affect the heart. Hydrogen ions, well, that's part of the buffering process that controls, well, what aspect of the blood? pH, yeah. So pH is going to be influenced by hydrogen ions. So, you know, by secreting hydrogen ions, you can reduce the acidity of the blood. You've then got ammonium ions, creatinine, and urea as being real serious waste products. And that's the bit where, you know, we could look at the production of urea by a process called the ornithine cycle that goes on in the liver. We're not going to. We're not even going to do it in the biochemistry unit. There are certain things that, you know, the biochemistry of that it's not necessary. It doesn't help you particularly. But urea just comes from old amino acids, and it's the nitrogen bit that's tricky to get rid of. And you can see nitrogen there in the ammonium ions as well. So those are real waste products. Also, of course, hormones. Well, hormones have to be present in the body in differing concentrations to achieve the control process, so they're constantly removed by the kidneys, deactivated by the liver and then removed by the kidneys. And similarly with drugs, I've put e.g. penicillin, but it could be e.g. pretty much anything, you know, paracetamol, anything that you like really is going to be deactivated by the liver, and then the kidneys will get rid of it. So, you know, that's not an exhaustive list by any stretch of the imagination, but it just gives you an idea of some of the some of the substances that we actually need to excrete. And I think as far as content is concerned for this morning, that is enough, really. And it will leave me with a little bit of recap and summary to do next time, because obviously we're missing a few people. And yeah, it's absolutely, yeah. With all medication, you have to take it perhaps every 4 hours, every 6 hours, twice a day, whatever. And it's because your body is constantly getting rid of it. So otherwise, you'd only need to take one, and it would do its job. It would stay in circulation. But it's also, well, that's it. But that, yeah, it's not the effects wear off as such, but it's taken out of the body. Yep. Yeah, so, you know, and you can obviously use urine as a test for drugs and certain hormones and things as well, can't you? So there you go.